Welcome to the first lecture of genetics. In this lecture, we're going to look at the father of genetics, Gregor Mendel, and some of the inheritance rules that he figured out for genetics and some of the groundwork that he laid for the rest of the world to continue on his work. His original work wasn't really well accepted when he first did it. It wasn't until 30 years or so after he did his initial research that people started to catch on to what he was, what his findings were from his research. All right, so Gregor Mendel was a monk, and I'm not going to test you on what he was, but he was a monk that started to ask the right questions and do the right research and laid some good groundwork for genetics and a lot of the main modern principles we still use today for studying genetics and for looking at how things are inherited from parents to offspring, and how things are inherited through a family, through different generations. And we still study new characters for different organisms, but we look back to these basic principles in order to figure out new principles. So what Gregor Mendel did was he wanted to know how certain characters, he called them, are inherited. So, for example, like seed shape. Okay, that was a character. And then there's a variety of types of characters, which we call traits, like oval seed shape, square seed shape, triangle seed shape. Those would be traits. Now, again, you're not going to see a triangle seed shape, but those would be the traits. So, character refers to the broad category, and then traits are the different types. Okay, so he wanted to know, how are these characters inherited? Why, if this parent creates an offspring with this parent, does the offspring look a certain way? Okay, and he used pea plants. He used garden pea plants and studied their characters. He looked at things like seed shape, seed color, flower color, or petal color, height, width, a variety of different characters, which you can see in a chart in your textbook. And he came up with a variety of things because of that. He was able to figure out some basic rules for looking at inheritance patterns in organisms. Okay, and we're going to look at those now. So what he figured out was, first thing we're going to look at is there can be a dominant and recessive traits. Okay, dominant and recessive traits. All right, dominant meaning this is the one you're most likely going to see if it's present. If it's present, the dominant trait's going to show. Recessive is the one that's hiding in the background. Because what happened was, let's say we had a person who has, let's say we'll go with seed color here. So big Y is yellow, little y is green. Okay? So little y refers to recessive. The lowercase letter refers to recessive. The capital case letter refers to dominant. Okay? And what he figured out was he could take someone who had a yellow seed color and another parent who had yellow seed color and cross them, and he could get a plant with green seed color, which is two recessive together. And that's how you're going to get the recessive, is if two of them are together. If these two come together, you get recessive. Okay? So what he found was you have a dominant trait, which we'll come up with another term for that in a minute, but we have a dominant trait. When a dominant trait is in that genetics, or in the genome of that organism, it is shown. It is seen on the organism, is physically represented. Although they still could have the recessive trait, it isn't seen unless they have two of the recessive traits, one from mother, one from father. So that's how we outlined it as recessive. Now, there are some characters, traits in society that are recessive that you see as the majority. For example, 5 fingers is a recessive trait. But that's what you see as a majority. There's disorders that are dominant traits, like Huntington's disease is a dominant uh, trait. So there's some. It doesn't necessarily mean it's the one you always see. Dominant just means if it's there, it's going to be physically represented by the organism. Okay, so we figured out there was dominant and recessive traits. Now, in today's world, let me clear this ink, we call those alleles. Okay, we would say dominant and recessive, so D and R, alleles. Now, what are alleles? Alleles are, just like traits are different types of characters, alleles are different types of a gene. So, for example, a gene, in your body you have a gene for blood type. Okay, so you have a gene on your DNA for blood type. You have different alleles for that. You could have a blood type A allele, blood type B allele, blood type O allele. Okay, those are the alleles. You can have dominant and recessive alleles. Gregor Mendel figured that out just by doing research on his pea plants. Now, there's also some other terms that we need to know here. If you have two dominant alleles together, so let's say we have big D, we call this homozygous. And because they're dominant, we call it homozygous dominant. Okay? If it's big D, little d, so you have the dominant and then the recessive, you'd call this heterozygous because there's two different alleles. Okay? Up here you have the same alleles, different alleles, and then if you have two recessive, it would be homozygous recessive. So again, the term homo referring to the same, okay? Two of the same alleles. Okay, so make sure we know those terms when we're looking at genetics, because that'll be important. Some other terms we need to look at: genotype, and phenotype. Genotype is the genetic makeup of an organism. For example, if I had type A blood, okay, my genetic makeup could be, well, I have an A allele and an O allele. I show A blood, but I have an O allele in there. O is just recessive. I only see the type A blood, but my genetic makeup is both of these. I got one from my mother and one from my father. So that's my genetic makeup. My phenotype is the actual physical expression you see. When you look at a person or when you look at their blood, what do you see? So in this case, we would say, well, it's A blood. But in the genetic makeup, we also have that O allele. And that's how things can be not seen in one generation and then seen in the other, which is what Gregor Mendel found out when he was looking at seed color. He had one generation that was all yellow and then the next generation that had some green. And he was able to figure out, well, there must be these recessive alleles that are hiding out, and then they show up in later generations. What Mendel also figured out was that there was two main principles, which we call them laws now, that he figured out that help us lay groundwork for modern day genetics. The first one is the law of segregation. Okay, so segregation. And if we just look at the term segregation, what does that mean? It means to separate, to divide, segregate. You're going to divide. You're going to divide something. Okay. Well, in this case, what he figured out was that a person has at least two alleles or a pair of alleles for one gene. Okay. For example, blood type. Let's talk about blood type again. Let's say from my mother I got the allele A, and from my father I got the allele O. Right? That's in my DNA. When a person goes to make sperm or eggs for fertilization, these are going to separate, right? If we think back to meiosis, the chromosomes separate. Well, this is going to be on a chromosome for my mother, and this one's going to be on a chromosome for my father. The chromosomes are going to separate, and when chromosomes separate, so do the alleles, right? Because they're found on the chromosome. That's where genes are. Genes are a section of DNA that code for a specific protein. Okay? They code for a part of our body. Alright? So the law of segregation says, well, when we go through meiosis and we create gametes, which are your sperm and egg, the alleles separate. So one of the sperm or egg will have O, and one of the sperm or egg will have A. So depending on who fertilization occurs with, you could get blood type B, blood type O, or blood type A with this genetic makeup. Okay? The other one is law of independent assortment. And we've seen this term before, and they are associated. Okay? Law of independent assortment says that characters found on different chromosomes will be inherited in different ways. Okay? There's no specific way chromosomes lined up or genes on different chromosomes are associated together. It's a law of independent assortment. Seed color and seed shape aren't inherited together necessarily. Okay? If they're on different chromosomes, they can be inherited differently. Okay? There's no dependence between genes. All right? It's all up to the chromosomes and how they separate. So those are the two laws that we have from Mendel's research. So let's go back here. Let's clear this page here. So let's look at how we can actually complete genetics and how Gregor did it, Mendel did it when he was doing his research. So let's just look at seed color, okay? So let's say Mendel was looking at seed color, and he had one plant that was homozygous dominant and another plant that was heterozygous, okay? And he was going to fertilize, okay? He was going to take the pollen from this plant, the seed from this plant, and fertilize that plant, okay? Alright, this is what we call, these are the parents, okay? These would be our parents, the parent generation. These are the two genotypes we're crossing, okay? This would be a genotype here, and this would be a genotype. Alright, we're going to cross those. So first thing we need to do, and this is how you'll do it for all genetic problems, is we need to figure out if we're going to cross these, what are the possible gametes, or sperm or egg, that these genotypes would make. So let's first look over here at parent 1 right here. It's parent 1. The the parent 1 genotype has a dominant allele and another dominant allele. So when they separate due to law of segregation, one of them is going to go to a cell and another one's going to go to a cell. So the possible gametes are big Y and big Y. This one will be in a cell and this one will be in a cell. Now there'll be other these are just on a chromosome, but we're just looking at this one allele and gene. Okay, so that's the possible gametes for that one. Over here, the possible gametes, if we separate these, are big Y and little y. Okay. Now once we figure out the possible gametes, we can plug them into what we call a Punnett square, which is what allows us to actually do a sample cross of the parents' genotypes and figure out the possible offspring. So let's look at the Punnett square here. I'll put the possible gametes on this side over here. Now because there's two dominants, we don't necessarily need to do the second one. It's going to be the same thing. And over here, put the big Y, little y. Okay, so we actually won't even need to do this bottom part. We can, but we don't need to. And then what we do is we actually cross them. So we pretend fertilization occurred. So this sperm or egg over here fertilized with this sperm or egg. And when they come together, they create big Y, big Y. And then this one goes with this one, and when they come together, it's big Y, little y. So now at this point, we have the possible genotypes of the offspring. Okay, the possible genotypes of the offspring. And those possible genotypes are 50% big Y, so that's 50% there, and 50% big Y, little y. That's the genotypic ratio, we would say. Okay? It's a 1-to-1 ratio. Now, if we're looking at phenotypic ratio, if I clear this ink here, we had big Y, big Y, big Y, little y. The phenotype of this would be, we know big Y is dominant, and big Y refers to yellow seed color, so we would say yellow 50%. And down here, we know big Y is dominant and it's yellow, and here there's a little y and it's green, but it's recessive. And anytime you have a heterozygous and it's dominant and recessive, the dominant is going to be the one seen. So in this case, it would still be yellow. And so in this case, it wouldn't be 50%. We have yellow for both genotypes, so it would be 100% yellow. So the genotype is a 50%. You're going to get two different types of genotypes, but you're only going to get one phenotype. Okay, and so you got to be able to make that distinction between a genotype and a phenotype. Okay. Now the cross we just did there is what we call a monohybrid cross. Okay, and you're just doing one generation. Okay, this right here would be what we call the F1 generation. It's just one offspring generation. Now if we wanted, we could cross these two now, and we could get a F2 generation. So let's go ahead and do that. So we're going to take these and cross them, and notice they're the same one. And so instead, actually, let's do this. Sorry, let's do a self-fertilization of that one. So let me clear this ink here. So we're going to do a self-fertilization. So the two genotypes we're actually going to be crossing is two heterozygotes. So this was our F1 generation. These are our parents. We're going to get an F2 generation here. First thing we do is figure out the possible gametes. If we divide these, we're going to get big Y and little y. Same thing on the other side. Put them in the Punnett square. Big Y, little y, big Y, little y. Here we'll get big Y, big Y, big Y, little y, big Y, little y, and little y, little y. And this is where we would look at the different genotypes. Well, we can see here we have one genotype there. These two right here are the same genotype, and then another one there. So we have three different genotypes. So big Y, little y would be 50%. Big Y would be 25%. And little y would be 25%. Now if we're going to use the proper terms on this, so the proper terms on this would be for little y, we would say homozygous recessive. For big Y, we'd say homozygous dominant. And for big Y, little y, that would be our heterozygous. And again, you would figure out the phenotypic ratio as well. So you'd want to figure out what's yellow and what's green and what's the ratio. And in this case, it'd be 75% yellow, 25% green, so it's a 3-to-1 phenotypic ratio. Next, we're going to look at some other inheritance patterns and then a dihybrid cross as well. Alright, let's go ahead and look at how you would solve a dihybrid cross, or when you have two factors you're looking at, or two characters we're looking at. So let me clear this ink here. So let's say we want to look at seed color and seed shape. So let's say big R equals, we always want to figure out the letters first, assign a letter. Big R equals round, okay, so round's dominant. Little r equals, let's say oval, okay, and big yellow again is, big Y is yellow, and little y is green, okay. And we're going to look at what we call a dihybrid cross. So let's say one parent has this genotype. So the parent is heterozygous for seed color and heterozygous for seed shape. And we're going to cross that parent with a homozygous dominant for color and shape. Okay? So after we do that, these are your first steps. That's step 1, assign letters. Step 2, get your parents' genotypes. Step 3, figure out the gametes. So this is where the gametes get a little difficult, okay, when the dihybrid. This is where people tend to struggle on figuring out the gametes. So again, law of segregation, right? Little y and big Y are going to split and go separate ways. Big R and little y are going to split and go separate ways. But due to the law of independent assortment, we know which way they're going to go. So big Y could end up with big R. Okay? Big Y could also end up with little r because we don't know which way the y's and the r's are going to go. You could also get little y, big R, and little y, little r. So those are your 4 possible gametes for that parent. Now we're going to look at over here. On this side it's a lot easier because they're homozygous. It's going to be big R and big Y. There's only one possibility. Because you could sit here and do this, but it's the same thing over and over again. Okay? And then what we're going to do is plug these right here into the Punnett square. So let me go ahead and clear the ink. And we'll make our Punnett square here. We may have to adjust that. So let's just do this. Big Y, big R, big Y, little r, little y, big R, little y, little r. And then there was only one, which was big Y, big R. So it's more like a Punnett rectangle because of the number of gametes. And then we cross them. And you want to make sure you keep the same letters together because they correspond with a certain gene or a certain character. And you want to be able to figure out what the phenotype is. So you want to see the interaction. So big Y, big Y, big R, big R. So this is one possible offspring right here. Again, these are possible offsprings. They could be any of them. These are just the percentages. We're figuring out the probabilities. Okay? Here we have big Y, big R, little r. Here we have big Y, big R, and down here, big Y, big R, little r. And now you again would figure out your genotypic ratio. Well, we have 4 different genotypes here, so each one would be 25%. 25% there, 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 and there. That's your genotypic ratio. Phenotype, there's actually only one phenotype here. And it is yellow seed color and round shape. So you would say 100% yellow and round. And that's how you would solve a dihybrid or double factor cross. Okay, if there's 2 different genes together, that's how you would solve it. Okay. Next, we're going to look at a couple different inheritance patterns that are variations off of what Gregor Mendel figured out. He figured out the dominant recessive, and after years of continued research, they were able to figure out some other inheritance patterns. Okay, and one of them being what we call incomplete dominance. Incomplete dominance. And there's a great example in your book on red flower and white flower, so that's what we'll look at, the snapdragons. And so what happens in incomplete dominance is you get 2 phenotypes that come together and they create a new phenotype or a blended phenotype. But you can't recognize the originals, and so that's what we'll look at here. So first one here is, we'll say we have a big R equals red, little r equals white. Okay? But when they're together, they equal pink. Okay? So when you get a heterozygote in incomplete dominance, it's going to have a different phenotype, and it's what we call an intermediate phenotype. This would be an intermediate phenotype. Intermediate phenotype. Okay? You can't see the red or the white, it's its new color. Alright? So let's go ahead and look at the actual problem here. So let me clear the ink on this page. So let's say we have a red snapdragon, homozygous dominant, with a all white snapdragon here. Okay? And now it's incomplete dominance when they're together. Right? Incomplete dominance when they're together, which means nothing's really dominant. There's not a complete dominance. Okay, and that's why you get the intermediate phenotype. So that's step 1. Step 2, figure out the gametes, big R and little r. Put them in a Punnett square. With this, your only possible outcome would be big R, little r. If you get big R, little r, that would be 100% pink. Okay? Now you could get, depending on the genotypes of the parents, you could get 1 pink and 3 red or 1 white, but it depends on the ratio. The ratio depends on the genotypes of the parents. Okay, that's these. But in this case, we wound up with 100% pink, 100% of the intermediate phenotype. So in incomplete dominance, when you have the heterozygote for that gene, you're going to get an intermediate phenotype, and that is incomplete dominance. An example in humans is hypercholesterolemia, which is increased levels of cholesterol in the blood. Okay, if you have if you're homozygous dominant, you're normal. You have normal levels. If you're heterozygous, you're like a little higher, and then if you're homozygous recessive, you have really high cholesterol. You can have heart attacks as like age 2. Okay, so that's an example in humans that for you to learn about there. All right, next one we're going to look at is codominance. So in codominance, instead of having one dominant or the other, they're dominant together. An example of that is blood type, A, B, and O. And in blood type, A and B are both dominant, which is why you get AB blood. Okay, and O is always recessive. All right, so let's go ahead and look at a example problem of that. So let's say we have a parent who has, and I'm going to use the letter I with the blood types on top of it to keep it organized. Okay, so let's say we have a blood type like that. So the capital I refers to the dominant A and B, and the little I refers to O. Okay, so little I equals O. And we're going to cross that parent with a heterozygous for blood type B. Okay, so let's figure out the gametes here. That's step 1. Step 2, gametes over here are those. Gametes over here are those. Now we're going to put it in a Punnett square, and I'm going to clear this so I can put it in a Punnett square. So I got my dominant B, I got my recessive O, my dominant A allele, and my recessive O. And we're going to cross them. So we'll have A, B. And the reason I use I is to keep it organized. If you have a bunch of A, B's, and O's, you can do it that way. This is just how the book does it and how I'll do it for you. Over here we have A and O. Here we have B and O, and then 2 recessive. Okay, so again, you would also look here at your genotypic ratio and your phenotypic ratio as we did before. But where codominance comes into play is when you have these two together right here. That is blood type AB, and that's the codominance. Okay, they're not dominant over each other; they're dominant together. They're both dominant over O, but when they come together, they create a phenotype that shows both of them. That's different than incomplete dominance, right? Because incomplete dominance, it's an intermediate. In codominance, it's its own. It shows both of them. Okay, it's not something new. It shows both of them. It's like, think of like something striped. You can see both colors. In blood type AB, you can see A and B proteins on the cell. Okay, and then you would have A here, B here, because they're dominant over O, and then there you have homozygous recessive, so that would be blood type O. Okay, so that is the end of part 1 genetics lecture. Okay, I know it was a little longer. The next one won't be as long. Make sure you understand the principles of inheritance, how you solve these problems. Okay, make sure you go down the steps. All right, and we'll practice these in lab, and we'll do a lot of genetics problems. So make sure you watch part 2 genetics.